Hi, my name is Lilian, and today I'm going to present a machine learning regression model for prediction and analysis of potential SARS-CoV-2 inhibitors. To enter the host cells, the coronavirus binds to a cell surface receptor known as AC2. A small segment of the virus spike, the RBD, is responsible to this attachment. Here we have a representation of the polar contacts between the amino acids of the RBD and AC2. We can reduce viral contamination by introducing small molecules that have a high affinity for the RBD and that will complex with it, preventing the link between RBD and AC2. The molecular docking technique can be used for screening of potential ligands. It is used to predict how strong a protein or enzyme interacts with small molecules, the ligands. Here, the target is the spike of the coronavirus. This figure shows an example of the ligand binding the RBD of the spike. The molecular docking is one of the fastest tools for screening of potential ligands. Even so, a personal computer can take months to analyze many ligands. Additionally, in general, molecular docking works do not study the properties of the ligands that influence the binding energy with the spike. We developed an artificial intelligence tool for faster identification of potential SARS-CoV-2 inhibitors and to help understand and identify the inhibitors features that most contribute to the best scores. The binding affinity of the spike with more than 90,000 molecules were used to train and validate a model of random forest regression, which can be used for supervised machine learning regressions. This procedure permits to reduce the time of simulation from weeks to seconds, while maintaining a good representability of the results. 18 filters of the ligands were analyzed. This figure shows in descending order the filters with greatest impact on the binding affinity between the spike and the ligand. Higher number of rings and heavy atoms favored the connection, decreasing the binding affinity, while increased the fraction of carbons that are sp3 hybridized and the number of rotatable bonds had negative effect on the result. The other filters had smaller influence on the output of the model. Our model accelerates the estimation and allows to evaluate the effect of the ligand structure on the binding affinity with the spike. Thank you.